Hello to our friends joining us via recording. We are going to wrap up our discussion of the Central Dogma. Before I flip over into kind of the region we were at last time, I want to remind us that when we talk about the Central Dogma, it means we're starting with our DNA and we're using our DNA to build proteins. What we talked about in class on Monday was turning that DNA into RNA. Oh my. I'm getting my notice that the school's closed this evening. Sorry about that. So what we talked about in class on Monday, turning that DNA into RNA. Can you help me out in the chat? What would be some reasons that we need to do transcription? Why do I need to turn the DNA into RNA? Why is that important? Did you all hear that? My computer's talking to me. Yeah, so Cassidy has for us one of the reasons. One of the reasons is that we need RNA to leave the nucleus. DNA can't leave the nucleus. RNA can leave the nucleus. The other reason that we're going to want to have RNA instead of DNA is RNA is much more specific than DNA is. Remember from our analogy that we talked about at the end of class on Monday, how Encyclopedia Britannica is like your DNA. All the information you could possibly need is stored inside Encyclopedia Britannica. But when we're writing a book report, we don't want all of that information. We just want information related to our particular topic. So when we look at the Central Dogma, not only do we need something that can get out of the nucleus, we also need something that's specific to what we want to build. So a quick reminder overview for us of the process of the Central Dogma. We're going to jump in right here, going from RNA into proteins. So whiplash, bear with me as we thumb through fast. All right, we are to the point where I have a molecule of RNA. That molecule of RNA leaves the nucleus and goes out to my organelles called the ribosomes. Remember that ribosomes are protein builders, but ribosomes cannot build proteins by themselves. Ribosomes have to work with that other kind of RNA that is called transfer RNA or tRNA. When we talk about the way that ribosomes work, there's a few important terms for you to keep in mind. So one of those terms I need you to underline, highlight, star in your notes packet is the term codon. Underline, highlight, star, codon. Ribosomes are not very good at reading RNA. They have to read it in these short little 3-letter segments that we see here. So these short little 3-letter segments, these are codons. When a ribosome is trying to build a protein, it's going to build that protein 3 bases at a time. So when a ribosome gets a codon, when it gets a set of letters, it reads them. So let's say the letters, we're going to pretend the letters are A, U, G. So the ribosome reads these letters, but ribosomes aren't very good at understanding what they're reading. So the ribosome calls out to the environment, hey, can anyone tell me what AUG means? And while it's calling out this AUG pattern, one of the nearby tRNA molecules, which remember from our discussion last time, are attached to amino acids, that tRNA molecule says, oh yeah, I can tell you what that means. I can tell you that that means we need the amino acid methionine. So that tRNA molecule comes over to the ribosome and says, here's the piece that you're looking for. Here's the amino acid you need. Ribosome says, perfect. Now I'm going to read some more. It goes to the next set of 3 letters, maybe C, C, C. And the ribosome calls out to the environment, hey, who can tell me what C, C, C means? And a new tRNA molecule that can understand that will come over and say, yeah, here's the amino acid that you need for that sequence. So the way that a ribosome is going to translate or turn RNA into a completely new language, that language being a protein, is it uses this thing called tRNA. As the ribosome goes through and reads the RNA and keeps adding these amino acids, it puts together those amino acids using that special kind of chemical bond that we only find in proteins. Can anyone remember the name of that chemical bond that we only find in proteins? What was that special one just inside proteins for my primary structure? Exactly. Yep. So my primary structure is built with peptide bonds, and peptide bonds are built by ribosomes. So ribosomes make my peptide bonds to connect each of these amino acids together to build us a protein. So when we're looking at the process of translation, we started with the mRNA molecule that was made for us using transcription, using that enzyme called RNA polymerase. This RNA molecule gets to the ribosome, and the ribosome looks for a codon. It looks for the set of 3 letters. It reads those letters. It says, hey, who understands this? And the tRNA molecule says, I understand this. Here's what you need. Ribosome says, perfect. Who understands this one? This tRNA molecule says, I know what you need. Here it is. So it goes down this mRNA strand, reading it in codons, getting all of the information that it needs to build a protein chain. When we talk about codons, there are 2 really important kinds of codons for you to be familiar with. Important kind number 1 are these ones we see right here, start codons. Now, there is only one start codon. It's always the same across all proteins. The start codon is AUG. When a piece of RNA gets to the ribosome is frantically looking for AUG because that's how it knows where we start the process of translation. So the ribosome finds AUG, it calls that out to its friends tRNA, and the tRNA molecule that can understand AUG brings over the amino acid methionine. Once we've added methionine, every single one of your proteins start with methionine, by the way. Once we've added that, then my ribosome can move on to the next codon. And it'll read that one and that one and that one. It's going to go all the way through the chain, but it always has to have that start codon. We always have to start somewhere. The other kind of codon that's really important for you to be familiar with is the stop codon. So the stop codon is a codon that when we get to it, there are no tRNA molecules that understand it. So the ribosome calls out to the environment, UGA. Who can tell me what UGA means? Nobody responds. Ribosome tries again, UGA. Who can tell me what this means? Nobody responds. Tries one more time, and when no one responds, the ribosome literally falls apart. Ribosomes are made out of more than one piece. So when they can't translate something, when nobody can help them read it, they're literally going to fall apart, and the process of translation is going to end. So stop codons are really important because this is how your cells know to stop reading the message. My protein is fully built. Now, here's an important idea related to stop codons. We always want our stop codons to be at the very end of our RNA strand. We can get into trouble when my stop codon ends up earlier in the RNA strand. So let's consider something together. Let's suppose that RNA polymerase did a bad job and it accidentally put a stop codon right here. Let's pretend that we put a stop codon here. Instead of this codon, we put a stop codon here. What effect do you think that might have on the protein we're trying to build? If I put a stop codon right here instead of this codon number 4, what do you think might happen? So let me get a pen. Instead of being number 4, Now we got a stop sign. What do we think? Okay, so several of us are bringing in one of our protein words we talked about, denaturing things. Yes, so if something's denatured, it doesn't have the right shape permanently. Although with denatured, we had to start with the right shape. So we're halfway there. We're picking up on my shape is wrong, right? I like the way that Hayden mentioned to it, and Max as well. We would build a completely different protein. Because remember, at a stop codon, we're like, oh, this is the end. I'm done. So I put a stop codon in right here. Everything after this point is going to be ignored. I'm not going to read it at all. So I was supposed to put in 9 amino acids. Instead, I only put in 3. And we all already kind of hinted at this, but if I put a stop codon in early, do you think I could or could not still do my job? If I put that stop codon in early, do you think that protein is still going to be able to do its job? Yeah, it definitely could not still do its job. When we put in that stop codon early, that wherever I put it in, that is the end of the line. That is the last amino acid I'm putting in. So when we're looking at sequences of DNA, or of RNA, excuse me, we really hope that I don't have a stop codon in too early, because if I do, my protein stops being built right there and it doesn't go any further. So every protein has a start codon and everyone has a stop codon telling us where to begin and where to end. When we take quizzes or when we do the homework or we do an exam, I will ask you to translate an RNA sequence for me. So I'll give you a set of letters and I'll ask you, what amino acids are we going to put in a chain? When I do that, you can't just call out to tRNA, hey, tell me what this codon means. You have to use the table that we see here on the slide. So I'm going to type for you. This table is called the genetic code table. I promise you, you don't need to have this table memorized. I will give it to you, but you do need to know how to use it. So let's make up an RNA sequence over here. By the way, if I'm ever asking you to do translation, I will give you a DNA sequence or an RNA sequence or a DNA sequence to get you there. So I got the question earlier today, where did that come from? Well, I would give it to you on an assignment. Whenever you're doing translation, the first thing you do is we split stuff up into codons. So we got to chop it up into these little sets of 3 letters. Every time you do translation, we start there. Then we go to our table. So notice over here on the left, my table has a side over here for the first letter. So the first letter on the table, the first letter in my codon is an A. I know that the codon I'm looking for is somewhere in this row. My second letter in the codon is what's across the top. So my second letter here is a U, which means that I'm going to be in this column. So now I know I'm somewhere inside this box. You can find the third letter over here on the side, or you can just look inside the box. My first amino acid that I'm putting in for my first codon, AUG, that causes me to put in the amino acid methionine. Now that I've translated one codon, I'm going to move into my next codon. So this one starts with a C. Now I'm in this row. And my second letter is a C. Now I'm in this column. So I'm somewhere inside this box right here. We are CCA. That's going to give me proline. Hey, now check this out. When I got to the box, let me get my pointer back. When I got to this box right here, notice that it didn't matter if I had an A at the end or a C or a U or a G. It didn't matter what I had at the end. I was automatically going to get proline. The fact that all of these different codons leads to the same amino acid, this is another underline, highlight, star word for you. I'm going to type it in the chat. It's called degenerate. Degenerate. The genetic code, the way that your cells make proteins, it's a degenerate code. What that means for us is there's wiggle room. Say when RNA polymerase was building the RNA molecule, it got a little bit sloppy. So it was supposed to be CCA, but RNA polymerase built CCU instead. Well, the good news is my ribosome reads both of those the same way, CCU and CCA. Now, if my ribosome got sloppy and instead of CCA, it did CAA, I do or do not get the same amino acid. Can we tell? Is CCA going to give me the same one as CAA? Uh-oh. Can you all hear me? I'm getting no responses. We said that CCA gives me proline. If I do CAA, it's giving me glutamine. I get a different amino acid if I change that second letter compared to if I just changed the third letter. And the same thing happens if I go next door. If I did CUA, I end up with something different as well. And if I go totally crazy and change it from CCA to UCA, completely different amino acid. When I talk about being able to have some room for errors, which is what degenerate means, I've got room for errors. I only have room for errors in the last base in my codon. Only the last letter can be changed. I have a special name for that last letter in a codon. I put it in the chat for you here. It's called a wobble base, as in wobble baby, base. If we were in class, you could see that Dr. Ellis cannot wobble, but I like to pretend. The wobble base is the last letter in a codon. I can change the last letter in a codon, and I might still be okay. I cannot change the second letter. I cannot change the first letter. I can only change the wobble base. So my code being degenerate means that more than one sequence might get me to the right amino acid. But the caveat with that is that extra sequence that might get me there, that extra sequence has to be different only in the last base pair. How do we feel about the table and about this idea of a degenerate code? Thumbs up, or what questions do we have? Awesome. Okay, lots of thumbs up, lots of goods. For the sake of time, I'm not going to take class time to translate this with you. But let me remind you, the first thing we do every time we're translating, we chunk up our RNA sequence into groups of 3. So when you're working on this at home, please do work on this at home. Start by chunking up our codons into groups of 3 letters. Then use that genetic code table to help you fill it in. I'll mention, again, for the sake of time, I'm not going to sit here and show you a video on my screen so you can watch it on your screen. In the guided lesson, there's a really awesome video that shows you 3D imaging of how the process of the Central Dogma works. So from opening up your DNA to building RNA to turning it into a protein, all of that is shown, and not just in pictures. It's shown the whole process. please make sure to watch that video in the guided lesson. It'll help you to put these steps in order. So the steps of protein synthesis that we see here, from where we start with RNA polymerase, making RNA, all the way to the end when we finish making our amino acid chain. Yeah, so Angelita agrees. This video is very helpful for you. So please take some time to go through and watch that. What I want to talk with you about before we dive into group work is what happens if I change something, but it's not the wobble base. So what we're going to talk about to help us understand why the wobble base is the only one that can change is when we're looking over here at a condition called sickle cell anemia. So sickle cell anemia is a condition in which people's red blood cells look like a sickle. So you know how we've been drawing all those salty banana cells? We've kind of been drawing sickled red blood cells. So a sickle is something you can use to harvest wheat. It's got a blade like this and a little handle. So that's why it's called sickle cell. Sickle cell anemia, instead of my red blood cells being these flat circles, like I see here, my red blood cells are shaped funky like this. Hey, not a trick question. If I'm shaped like this, am I going to be able to do the jobs that this one does? What do we think? Yeah, I am definitely not. Y'all got my question, right? This is my question from like every part of our lecture. If I'm shaped like that, if I'm funky shaped, I can't do what a normal red blood cell can do. So let's talk about how we end up with red blood cells that look like this instead of their normal round circles. It all starts with the DNA. It starts at the very beginning of the central dogma. If I do not have a mutation for sickle cell anemia, the sequence of my hemoglobin gene, I should mention that we're talking about the hemoglobin protein. The sequence inside my gene includes CTC. That's my normal sequence. So when RNA polymerase builds mRNA, that's the complement to this, I make a codon GAG. When I make the codon GAG, that puts in an amino acid called glutamic acid. That's normal situation. Say, though, that I have a mutation in my hemoglobin gene, and now I'm putting in CAC in my DNA. When RNA polymerase reads that sequence, now it's going to put in GUG. GUG, we have a change, not in the wobble base at the end, but in the middle. And because I have a change in the wobble base in the middle of this codon, I end up putting in a completely different amino acid. That amino acid is valine. When we talk about accidentally adding the wrong amino acid to our protein, we have to go back to what we talked about with our amino acids. Remember that our amino acids have 4 different personality types. If I put the wrong kind of amino acid into my protein, but it's still the same personality type, maybe my protein will be okay. Maybe it won't matter, it won't be a big deal. So if I went from, oops, I lost my pointer. If I went from glutamate, which is what we started with, if I somehow mutated it and I ended up with aspartate, hey, we're the same personality type, we're both negative, we're good. If I went from glutamate to histidine, these both still have a charge, so one's positive and one's negative. Things that are positive or negative, do they play nice with water? Do they like to be around water if they're positive or they're negative? What do we think? We're divided. Think about my ionic bonds. When we talk about ionic bonds, remember that they fall apart in water because ions things with a positive charge, they love water. So if I go from a negatively charged amino acid to a positively charged amino acid, I mean, they both like water, so that's okay. Or same deal with my polar amino acids. Remember, polar things also like water. But when we talk about non-polar things do or don't like water. If I'm non-polar, do I like water? Exactly. Yeah, several of us chiming in. If I'm non-polar, remember, non-polar things are like, I don't have time for that kind of negativity in my life. If you are water and you have dipoles, stay away from me if I'm non-polar. So Max totally gets where we're going, right? So we started over here with this. We started with glutamate. Glutamate is totally chill and fine with water. We went to valine. Valine is nonpolar, meaning valine is not totally chill with water. Valine is hydrophobic. So we're going from the DNA, now we're looking at the protein side of things. Remember, I told you we're doing hemoglobin. We're building that oxygen transport protein inside our red blood cells. So we had our DNA sequence that had an error in it. We used that error to make a bad RNA molecule, and then we turned that bad RNA molecule into my primary structure, into my order of amino acids. So normally, we've got the glutamate right here. Now I put in the valine. If I go from a negatively charged amino acid to a non-polar amino acid, that's going to mess up the way that I make alpha helices and beta sheets. That's going to mess up the way that I fold into a 3D shape. My secondary and tertiary structures are going to be messed up. Normally, if I have the right amino acids, see how I've kind of got this heart-shaped protein? When I have the wrong amino acid in there, when I added valine, I end up with something, I think this kind of looks like an elf's shoe over here, like at the North Pole. The shape maybe matters a little bit less than this. I need you to underline, highlight, star this right here. When I put in valine, valine is hydrophobic. Valine does not like water. And if I put valine in there, I end up with this little spot because I can't fold correctly. I've got this spot right here that's hydrophobic. Remind me, hydrophobic. I do or don't like water. Hydrophobic. Yeah, exactly. I do not like water. I hate it. I'm afraid of it. Absolutely. So when I put the wrong amino acid in there, I end up with this space that is afraid of water, that hates water. So I take this protein that I built, one of the pieces of hemoglobin, and I put it together with the other piece of hemoglobin. So this other piece of hemoglobin is totally fine. Alpha is totally fine here. We build our quaternary structure where we have more than one chain together, and I definitely look different. Here's normal. There's not normal. Where it gets really dicey is when we start putting these hemoglobin proteins by each other. In normal circumstances, hemoglobin proteins stay alone. They each exist by themselves. But if I've got this big hydrophobic area where I don't like water, and my neighboring protein has this big hydrophobic area where it doesn't like water, remember how we talked about in a previous lecture, those things called hydrophobic attractions. Hydrophobic attractions. I don't want water here. You don't want water there. If we squeeze together and attach to each other, we can get rid of these places where we don't want water. We'll squeeze it together and push the water out. So instead of having little individual hemoglobins that are floating around inside my red blood cell, what I end up with, if I have the sickle cell trait, is I end up with hemoglobin proteins that are all attached to each other in these big long lines. And when they attach to each other in these big long lines, they switch my cell out into all these funky shapes. So it goes from being this nice round flat cell into this cell that's stretched into this sickle shape, because all of my hemoglobin proteins are clumping together instead of just floating around like we see here. Hey, not a trick question. Is this red blood cell going to function as well as this one does? Is this one going to do its job like this one does? Yeah, absolutely not, right? There are two things that these cells need to do for you. Number one, they need to transport oxygen. I need my hemoglobin proteins to be separate from one another to be able to attach to oxygen. So when I clump them together, I can't attach to as much oxygen. The other thing, number two, that these red blood cells need to be able to do is to squeeze into tiny places throughout your body. Your smallest blood vessels are really, really small. So I need to be flexible. I need to be squeezable. Normal red blood cells are that. Sickled red blood cells, not so much. They're really rigid. They're really stretched out. Sickled red blood cells are really good at making clots, at blocking places. So in individuals with sickle cell anemia, they have these things called crises, where what's happening is I actually stop sending blood to part of the body because all of these red blood cells are stuck on top of each other. They can't squeeze through the hole. So a patient in a sickle cell crisis is literally feeling the pain of the tissues in their body like dying because they're not getting oxygen and nutrients. The good news is we've actually started to use gene therapy to treat sickle cell anemia. So if you're interested in learning more about sickle cell anemia, there are actually several articles in the Current Events and Anatomy library that talk about sickle cell anemia. There's a patient named Victoria who volunteered to be in a clinical trial where they actually changed her DNA to help her start making red blood cells that are not sickled. So if you want to learn more about this, it ties in very well to what we're talking about here with the Central Dogma. If I have a problem with the DNA, I'll have a problem with the RNA. And if I have a problem with the RNA, I probably will have a problem with the protein as well. And that's what sickle cell anemia shows us. Yeah, so Lisa asked a good question that probably brings up content from other classes. The reason that the sickle cell trait hasn't just disappeared from an evolutionary perspective is if your red blood cells are shaped like this, it's harder for the parasite that causes malaria to set up shop inside of them. So it's harder for someone who has these sickled red blood cells to actually get infected with malaria, which is why that trait is much more prevalent in Africa, in populations where malaria is pretty common. You'll see people that typically will have inherited from one parent a trait, the sickle cell trait, and from another parent just kind of normal red blood cells. So we call that someone who has the trait as opposed to someone who has sickle cell anemia. Full-fledged sickle cell anemia, everybody looks like this. That's not good. Sickle cell trait, some of my red blood cells look like this. And if some of my red blood cells look like that, it's harder for me to catch malaria. So yeah, great connection to make with microbiology, malaria and red blood cells. Any other questions about translation before I split us up into our groups to complete our group work for the day? All right, well, I'm getting no questions, which means I'm going to go ahead and turn off my recording. Oh, I got one last question. That sickle cell anemia, it's caused by a mutation. Yep. Do we remember what gene we were having issues with? What are we making the wrong way with sickle cell anemia? I'll back up to that gene that was mutated. All the way back here at the DNA. If I mess up one of my codons in the DNA, yep, I mess up the DNA, then I make the wrong RNA. I make the wrong RNA and I put in the wrong protein, or the wrong amino acid, excuse me, and then my hemoglobin protein is shaped incorrectly. Yeah, so we start at the DNA level in the hemoglobin gene, and that hemoglobin gene has the wrong sequence, which means everything else in the central dogma is also going to be messed up as well. All right, I'm going to end my recording.